In this video, we will study about end replication problem, the role of telomeres and how it is related to aging. So we know during replication, DNA polymerase can only make the strand in 5' to 3' direction. So it makes strands in 5' to 3' direction, and the next is that it needs a primer which has free 3' OH group. So when leading strand has to be formed, we can see there is a primer which is attached at one of the ends, and with only one primer there is a free 3' OH group. The strand will be formed at one go without any interruption. But for the other strand, many primers are attached, and wherever there is free 3' OH group, there will be a strand formed here, then at the next, and then at the next. It means it will form in small fragments, which are Okazaki fragments, and that is why it is called lagging strand. After these Okazaki fragments are formed, the primers have to be removed. So now the primers are removed from wherever they are present. This end replication problem is mainly in the lagging strand, so we'll focus on this. So what happens here now, that these have to be replaced. So wherever these primers were present, there is a free 3' OH group, and so there is no problem for DNA polymerase to form the entire strand. This is 3'. But when the last primer is removed, which is present here, then there is no free 3' OH group present at this end. So it means if there is no free 3' OH group, DNA polymerase cannot form the strand. So the genes will be lost. So that is not the case. For that, the linear chromosomes or the linear DNA molecules at the very end of the chromosomes have certain structures which are called telomeres. Now these telomeres are repeats of TTA, GGG several times. Let's understand the role of telomeres in linear DNA molecules, and we are mainly talking about somatic cells. Somatic cells are also known as the body cells, which are present in the skin and heart, all the muscles. So when we look at this figure, we are mainly talking about the lagging strand and the parental strand which is attached to it. We are not looking into the leading strand because this end replication problem doesn't occur in the leading strand. So when the last primer is removed, this gap is left at the end of the lagging strand because there is no free 3' OH group for DNA polymerase to form the remaining strand. So what happens is, because there is a gap, no strand can be formed, so there is a overhang at the parental strand at the 3' end. So that is why what happens is that because there is an overhang, it has to be removed because it is extra. So another enzyme will come and it will cut this area. So gradually what is observed in somatic cells, that the telomeres start becoming shorter and shorter after every cell division. So here I have shown just one set of telomere. There are many. There are around 1500 to 2000 telomeres when a newborn baby is born. So gradually, as we age, with every cell division, these telomeres shorten when they reach a particular limit, which is known as Hayflick limit. This Hayflick limit was given by a scientist called Leonard Hayflick in 1962. So he said that after approximately 40 to 60 cell divisions, the cell enters into a Hayflick limit, where a signal is given to the cell that it shouldn't divide. After that, it reaches a stage of senescence, and these cells behave like zombies. They don't divide, but they stay. And they release certain chemicals which start damaging the adjacent cells. That is why we start aging and we have wrinkles, because there is accumulation of these senescent cells or these zombie cells in our skin. So this happens in somatic cells. So there is a difference in the germ cells and in the stem cells. Before we move further, let's first talk about telomeres a little more. So these telomeres are repeats of TTA-GGG. These are called protective caps. Why are they called protective caps? As mentioned, this is the telomere and it has a long length of telomeres, especially in newborns. They're born with around 1500 to 2000 these repeats of DNA. So what happens is these are called protective caps because they are the non-coding DNA. That means non-coding DNA means it does not code for any protein. So if they are cut, then there is no problem because they do not code for any protein anyway, and they protect the coding area which is present here. This is the coding area. So gradually when they are cut one by one, then still there is no effect because the coding area is still coding for the particular protein. When these telomeres are cut to a sufficient length or whatever number of divisions, 40 to 60 approximately, then the cell stops dividing. So for every division, around 30 to 200 base pairs are lost. So that is why they are called protective caps, because they are protecting the coding area. Now this telomere length is directly linked to aging. I've already mentioned that in newborn baby there are 1500 to 2000 these base pairs or repeats present. So gradually as we age, with every division it gets lesser and lesser, and by the time we get old, around 200 or 250 repeats are left. So it is directly linked with aging. A younger individual will always have longer telomeres than an older individual. There are some lifestyle changes which can increase the length of telomere and slow down aging. We will talk about these lifestyle changes in some other video. But still, telomere length is directly linked to the genetics. It means if your father or your mother have had longer telomere length, then the child will be born with a longer telomere length. And that is why if there are two individuals from different families of same age, they might look different. That is, one will age faster if they started with shorter telomere length. So that is why it is totally genetic, but still there are lifestyle changes by which we can increase telomere length and slow down aging. So let's move to the next topic. In germ cells, stem cells and cancer cells, they have an enzyme telomerase. So during replication, when the last primer is removed from here, then telomerase enzyme gets attached to the 3' end of the parental strand. It starts adding these repeats, that is TT, A, GGG, multiple times. So it has increased the length of the parental strand, so there is a overhang of this 3' end. After that, primase puts its primer. Now this is 5', 3'. As soon as DNA polymerase sees that the 3' free OH group is free here, it forms its strands. So the whole strand is formed, and then after that strand is formed, then the primer is removed. As soon as the primer is removed, then what happens is that whatever is extra has to be removed. Like this portion is extra, so this has been cut. But still the main gene is left, that is the coding area is left, but still the telomere length is not that short as compared to in somatic cells where there is no telomerase enzyme, and so there is no increase in the length of the telomeres. So this is the difference in germ cells, stem cells and cancer cells, where the telomere length can be increased. So these are in germ cells. Germ cells are like ova and sperm, and stem cells like in the bone marrow or the intestinal lining where telomerase is active. And then in cancer cells, the cancer cells can activate telomerase enzyme and then can multiply indefinitely. So the last point, that why germ cells or stem cells have telomerase activity and why the telomeres keep extending there, whereas in somatic cells telomerase activity is not there and they do not divide after particular number of divisions, or the telomerase do not extend. The main reason is because germ cells must remain immortal as they have to pass the complete set of genome to the next generation, whereas somatic cells stay mortal to prevent uncontrolled cell division that can lead to cancer. So in somatic cells, if there is any kind of mutation and if they are immortal, then they'll keep dividing and it will develop into cancer. That is why after particular number of divisions, somatic cells become senescent and then they stop dividing. So I hope you like my video. If you like my videos, then do subscribe to my channel and share and comment. Thanks for watching.